I can't imagine Monday. I mean, like I heard a lot from everyone, and I was like,
yeah.
I can't imagine being on a crutch in that situation.
It's been a pretty bad Monday, but ever since, like, it's been pretty good. It was nice, like opening doors, trying to get food in common. It's so, yeah, it really is a burden for me. Work and stuff. Man, get around pretty well.
You guys know that they have sushi today?
Get around.
Do you know what time it is?
Getting food in common, what a struggle.
It's gonna be gone when we arrive there.
I spent
But I work at 4 too at Olin Road, so we're covered 7, so I can go in.
next time.
Was it there will be no next
It's gonna be
time.
free.
It's gonna be free.
New perspective in life right here.
Yeah, that is C4 is kind of expensive.
Yeah, but I'll be really excited for the
Where else are you gonna
I suspect that probably I'm gonna like half off.
Oh, there's
there's
one thing
I'll probably like semi-crutching
something.
That's really good.
That's legit. That's legit too.
When you were around class and
I think it's owned
like
by
they I walk over and I'm like, well,
They have like, yeah, they have like like a little bit of
Short size, really good. Small. Yeah, it's just right down college. You can walk out.
Okay, you got here faster than I did.
It's small.
Yeah,
It doesn't mean it's automatically scanned.
Oh, why didn't you just ask Pat to
Okay,
bring
fine.
her
It's a
in?
little.
Just got here or
They
something.
have a really clean bullet though. Just as a little detail.
As much as I love him.
I like peppers.
not too bad.
Yeah, they're really spicy.
Yeah, I only have to come here one more time because I want to cast off.
Yeah.
Yeah, it's okay. It's not as spicy as it's
It's
supposed to
like 2
be.
weeks.
I wouldn't have a problem with
And
that.
even like also
But-
be a struggle getting here.
We have a pretty good restaurant,
Yeah.
Indian restaurant, like, like even the mild.
So what else are you taking?
It's proper
this
spicy.
but it's
Right.
They're
Before,
mild.
before that, I have multi.
Exactly. That's my problem with them. And I tried.
Yeah, it's pretty fun so
Call them
far.
up.
I'm
I
enjoying
talked to
it.
them.
I really like math, and
I'm
I
like,
didn't take it like this.
I'm good. Like
Pretty
it's
good
spicy,
but
but they still
I really do like it.
I went to Modern Surgery in December
And then
31st. Alone.
I'm taking English class.
The waiter's supposed to look at me and
I'm taking Montessori story.
she looked at me like,
Yeah, I think
Yeah.
what's going on?
Art Novella. I heard really good for that teacher stuff. That's really.
It's not really.
It was
Yeah.
like, you know what? First,
I mean, it's not really.
I wanted to originally get into the writer's one, but I tried to position it.
Ira Sean's back home.
That's one with the.
Yeah.
At that point, like the one thing was open was.
Oh, Taste of India?
So.
There's so many
Sure,
restaurants.
that's what I came up with, so.
We don't call it side.
But yeah, somewhere good.
Side.
I really, it's nice to like read a book that's not a textbook.
That is true, because I've been reading a book, Spicy in my class.
Like,
I also read a lot of those.
Are you gonna miss this?
Because it's
We're
kind
still
of
gonna miss
Oh, yeah.
That's not Dawn.
I got into knitting.
There's sushi at Chase Hotel.
That's good. That's
Yeah,
kind
it's-
of
I
had three
You should have asked for
Yeah.
Pero hay que saber de oposición.
Yeah.
Ahora es come back here.
I know.
Yeah.
You're productive and watch your show.
Yeah.
Good.
Thanks, Boots.
Yeah.
You like this class so far?
It's okay. I did some of the reading earlier. I mean, yesterday.
Yeah.
Interesting.
I did some last night and then this morning.
The first chapter.
This heavy last
First
night.
chapter was so good.
Love this.
That's last.
Really? What's your first one? What was
You don't need to read it yet.
Yeah. I was reading it. I was like, oh my god, 40 pages.
the time?
Like I'm not in the same research, but
It's just two.
and some people do.
Oh, that's like green.
It'll probably be, it's gonna be like raining though, so it's like.
So you
Yeah, it's it's bizarre. Like, it's climate change. It's fun. Sure.
Yeah. Even when they're silent,
Right.
He's the man. I miss him so
Colin's in
much.
here too. It's like our lab.
Yeah, it's like Sabati.
Party hard.
That really killed me. He's still here,
Okay,
but
like
like,
the
he's.
three of us had Delta lab together.
Right.
Just
Oh, I do have fun times.
I know some people are taking 270 right now on my Ecology and Evolution. I'm just,
That's like the worst ever.
I'm in 270.
you are, yeah.
In this room.
I just had to jump out of that class. I was like, maybe I'll take it next year, but I'm not interested in doing that anymore.
Wait, what are you majoring?
Well, I was thinking bio, but then I just didn't want to do 270, so I'll probably go biochem. Yeah. It was, yeah, I like chemistry a lot. And because I don't have whatever organic right now.
Yeah, I mean, you don't want organic to go to waste.
Yeah, I'll probably take inorganic at some point. And then, I don't know, some other chemistry.
I don't know. I just try to.
Yeah, but like I want to take,
Well, after this, you would only need one more elective.
and I probably want to take like biology at some point. Maybe bacteriology.
All of this. I don't understand how you're doing all of this.
Well, it's because I want to, you know? But like, if something opens up, I'll take it. Alright, a new class, like that's my goal. Yeah, I took this one, talked to somebody this, I guess,
Each page was
and
blue
he did it last
for no
semester.
reason.
That's why I changed it.
I don't know if it's better or not, but like, he said it was like 30 seconds. Oh, yeah, I usually say it's easy, so like, he said like, it was
No, I didn't take that time.
really?
That
Doesn't
is,
take time.
there's 8 slides. There's 8 good slides.
No, you do for the
Maybe.
slideshow.
No, there's 8 slides.
But I don't know.
I guess it
I don't
depends,
have
like,
that kind of time.
yeah, I went into organic, like, being pretty excited,
I don't have that
and like,
kind
so
of
far, I don't know, I'm gonna do that. I've been
I
enjoying
mean,
it.
I want to take.
I printed out as
I didn't.
but
I'm not going abroad next
they
semester.
printed out like one and they were single page. Like
like-
wasted like
Wait, they offer
I
it?
did that as half of my bio.
They offer
You
it
can do
every other
singles
year.
for
The
bio.
only time they offer it
Because
again
there's
is
like charts.
fall of March and May.
So we'd have to take it next year,
Yeah.
if I wanted to take it.
I just use it every
Nope.
year.
It would
What
take too
additional
much
stuff do we
time.
learn in advance?
Okay,
Like,
we're
what?
going to get started. There's
There's like
free sushi at 3 o'clock.
You just kind of
What? Where?
There is.
Chase.
Chase.
Can you all take a break?
How about we try to be done?
Okay, let's get started.
Okay. I'll do my best, but remember to, if you don't ask questions in class, send them to me by email. It's just easier in class because everyone gets to hear the answer. Any questions remaining from last time? I guess I'll probably, in the next day or two, I'll send out the groups. It'll just have to be random somehow, because otherwise it'll be too hard to organize. But hopefully, there's nobody that can't get along with anybody else in here, is there? Can we all just get along? Okay. We'll see how it works. If you have a major issue with your group, let me know, and I'll try to arrange something. But the first discussion will be 2 Fridays from now, so there'll be a bonus for whichever group gets ready quickly. Okay, so this class should be pretty straightforward. We threw a lot of big concepts at you in the last lecture, but it's just supposed to be big context. We went over everything we're going to talk about in the course, essentially, in the last lecture. So there's going to be very specific detail about each of those cell types, each of those issues. In this class, no problem. In this class, we're just going to talk about just some basics of the immune system physiologically, in terms of where in the body lymphoid organs are organized, and some basic means by which cells move around, and then just a quick medical example at the end. So, as we said last lecture, The primary lymphoid organs are the bone marrow and the thymus. I'll show pictures of them in a minute. The bone marrow is the location of hematopoiesis, as I said, and also development of a lot of the leukocytes, including monocytes and granulocytes, as well as B cells. T cells have a specific differentiation in the thymus where tolerance is maintained. And it's important to note that what comes out of the bone marrow, what comes out of the primary lymphoid organs are quote-unquote naive lymphocytes. In other words, ones that have not ever had an immune response. So everything after leaving the primary lymphoid organs is then a maturation, an immune response afterwards. So the process of producing naive cells comes from the primary lymphoid organs. The secondary lymph organs are the lymph nodes, spleen, and tissue-associated lymphoid tissue, which we'll detail in the upcoming slides. So the secondary lymphoid organs are the places where antigens and lymphocytes come in contact. I started talking about that last lecture in terms of the locations to which antigen-presenting cells travel to interact with lymphocytes. It's the primary site of the adaptive immune response, and the result is effector lymphocytes. So lymphocytes that come out of the primary organs are not ready to have an immune response. Ones that come out of the secondary lymphoid organs are ready to have an immune response. Okay, so again, bone marrow hematopoiesis occurs here. So maintaining all the white blood cell types, erythropoiesis, red blood cells, and the maturation and selection of B cells, which we'll talk about in a later class. So in terms of the number of cells in the bone marrow, the myeloid progenitors are the most numerous. That's not surprising because the majority of your white blood cells are neutrophils. They come from the myeloid progenitors, and they're turning over every 24 hours. So you're essentially, the life of a neutrophil is only about 24 hours. Then the erythroid progenitors, because red blood cells are so important and they also have a relatively short half-life. Finally, lymphoid progenitors, and the remainder are mast cells and structural cells. So again, there are nice diagrams. This is not important to memorize, but the basic structure is within the bone. I think we all sort of have a concept that, or anybody who's thought about bone marrow thinks that there's just a hollow cavity in there full of bone marrow, but it's actually a pretty elaborate structure. So on the outside of the bone is the periosteum. That's actually the living portion of the bone. Underneath that is the blood vessels, which supply the bone tissue. And they penetrate through the compact bone. That's actually the structure. That's the weight-bearing part of bones. And then inside the compact bone is spongy bone. And within the spongy bone is the bone marrow. So that bone marrow is in the central or medullary canal, which contains a red marrow. That's where all the erythropoiesis is going on. The yellow marrow, which is fat, or an energy source, essentially, for hematopoiesis. So basically you have, even if you're starving, you have this last source of energy within the bone to supply hematopoiesis. And then there's narrow spaces in the trabecular bone. So there's a one-directional circulation from the nutrient artery. So the nutrient artery penetrates through the compact bone into the bone marrow space, and then into the sinuses, and finally back into the nutrient vein. So just like other circulatory beds, there's an artery, a capillary bed, and a vein. And then within that space are all those different levels of differentiating hematopoietic cells, the original hematopoietic progenitor, as well as the common lymphocyte progenitor and the common myeloid progenitor. What leaves are mature myeloid cells, so myeloid being monocytes and neutrophils, and then naive lymphocytes. So the other component of the primary lymphoid organs is the thymus. And it has a very unique function in controlling the selection of T cells. So T cells that effectively leave the bone marrow have not actually been cleared for function. They could be autoreactive. So they have to circulate from the bone marrow to the thymus. And in the thymus, they undergo maturation to become able to undergo, hey.
Hi, you Professor Weaver?
No.
I'm sorry.
No.
Is this a,
Yeah, come in. Yeah.
Is this
This is immunology.
in kind of a daze?
How was the exam?
It was awfully long. I wouldn't recommend taking it if you don't have to.
Hang in there. Just let it wash over you. You're Cody?
Yes.
Okay. Welcome. The slides are on the website. Nabil is recording the lecture, so don't worry about it. So lymphocytes that come out of the bone marrow are not ready to have an immune response. They have to undergo selection and maturation in the thymus. So they travel through the blood system from the bone marrow to the thymus, and they have to undergo positive and negative selection. So as we talked about briefly in the last class, T cells require antigen presentation. So the T cell receptor is specific for some antigen, and that T cell receptor has been determined when the cell leaves the bone marrow. It reaches the thymus, and that T cell receptor gets tested. So positive selection basically says you have a functional T cell receptor. I don't really know what the antigen is to, but your T cell receptor works. It works well enough that you can get some positive signal through it, and I'll keep you alive. If you have a very dysfunctional T cell receptor that will never work to signal, you don't get positive selection, so the T cells get eliminated at that point. Then there's negative selection, which is even more important, which is basically the T cell cannot have affinity for a self-antigen. So somehow, almost any potential antigen that we express in any tissue at any time in our lives is expressed in the thymus. And if the T cell sees any of those antigens, it's eliminated. So you have to show that you have some functional ability to be positively selected, and you have to show that you don't react to any self-antigen to be negatively selected. So that process results in less than 1% survival of T cells. So 99% of the cells don't make those two thresholds, okay? And that process basically goes on through puberty, and then at that point you've essentially educated all the T cells you're ever going to educate. So you guys are about to be done. Yeah?
So the 99%, are they like this type or like do they?
Good question. So they undergo apoptosis. So just a quick, you guys have all had, actually I don't even know anymore. What are the prerequisites for this class in terms of biology? So basics, cellular, molecular, and intro biology. So somewhere in there you talked about apoptosis and necrosis. Okay, good. So this is a review. So essentially, non-functional cell death is necrosis. In that case, you get an enlarged nucleus, and you get destruction. You get a large cell size, and you get destruction of the nucleus. The membrane's disrupted. There's a leakage of cellular contents. Apoptosis is basically programmed cell death. So it's basically the cell has received a signal and it kills itself off, and that's a much more controlled process that involves shrinkage of the cell, specific digestion, and fragmentation of the nucleus, and clearance of an intact cell. So apoptosis is actually a negative signal for inflammation, whereas necrosis is a positive signal. So if there's a whole bunch of cellular debris, that's an inflammatory signal, whereas programmed cell death, where macrophages can take up an intact dead cell, is not inflammatory. So that's the process that's going on in the thymus with cells that are not effectively selected. So again, just necrosis basically results in inflammation because there's uncontrolled release. One of the mechanisms by which tolerance is maintained is by some antigens always being intracellular. So nucleic acids, et cetera, are protected from the immune system. When a cell bursts, all of those privileged antigens are exposed to the immune system, results in inflammation. Apoptosis results in uptake of intact cells and actually release of anti-inflammatory signals from macrophages. Okay, so the thymus is made up of T cells, a whole bunch of very specialized support cells that promote the differentiation and maturation of T cells. They have these specific names, but basically the idea is they supply all the important hematopoietic and growth factors for the T cells, as well as do that miraculous job of presenting all the potential self-antigens to T cells. It has, the physiology is not so important, but it basically has the structures I listed here, cortex, medulla, and associated artery and vein. So the thymus, and those of you that still have one, is located essentially between your lungs over your heart. It's a very thin tissue. It's organized by these into these cortical structures, and the specialized cells are described here. And again, the importance is not the structure, but the job it does, delivering those positive and negative signals. Any questions about primary lymphoid tissues? No.
Sorry, Aaron?
Yeah.
Why would someone not have a thymus?
Why would someone not have a thymus?
You said if you still have one.
Oh, because sorry, I said quickly in passing that essentially once you go through puberty, all your T cell education has happened, and so the tissue basically involutes, so it just becomes fatty tissue essentially. There are a few really rare diseases, athymic diseases, but usually that's because of some defect in T cells. It's actually formation of the thymus is T cell dependent as well.
Wasn't there a case where I guess they called the child bubble boy because he lived in a bubble because he was born without a thymus, so therefore he didn't have the immunity?
I know the case you're talking about. I think Bubble Boy had no lymphocytes at all. No T cells.
So it
or
wasn't
B
just the
cells.
thymus?
Not just the thymus. I'll have to look that up. I think Bubble Boy had. I'll look it up. It's a specific deficiency. I think it's ADA deficiency, which means you can't make T cell or B cell receptors. So they had no adaptive immune system at all.
Your body.
So T cell, when they do positive and negative selection, it's with both selections with MHC, or on PowerPoint it says negative selection. So it must have some affinity for MHC,
Right.
but then it has.
Sorry, I skipped over that. So, as I alluded to in the first lecture, the context in which antigen is presented to T cells is very important. And so, the T cell receptor doesn't only see the foreign peptide. T cells respond to pieces of protein, so short sequences of protein. And the T cell receptor has to not just see that non-self protein, but also self molecules, which is MHC. That's major histocompatibility complex. So that's another whole lecture on MHC, basically. And there are two major classes. There's class 1 and class 2. Class 1 presents self proteins, class 2 presents foreign proteins. But basically, the T cell receptor has to have some interaction with the MHC, has to provide some signal of interacting with major histocompatibility complex, or it's never going to work as a T cell receptor.
So for negative selection, the MHC will be self-produced.
So all MHC is self, and there's not all the foreign antigens in the world are not going to be expressed in the thymus. So the positive selection, it just has to have a little bit of function. It just has to be able to see MHC. It doesn't have to, the MHC doesn't have to contain the foreign protein. And what you're getting at is the fact that this is just hand-wavy. Nobody really knows exactly how it works. If you don't express, if in a MHC knockout mouse, the T cells don't undergo positive selection. So it has something to do with the interaction between T cells and MHC, but obviously the thymus doesn't contain all those foreign antigens for specificity, so it's just interacting with MHC. So the positive selection is basically your T cell works enough to see MHC and produce a little bit of signal to keep the T cell alive. Anybody else? Good questions. Okay, so primary lymphoid tissue, bone marrow, and thymus. Bone marrow produces most of the white blood cells of the innate immune system, and B cell development, which we'll spend a class on, happens there as well. T cells are not fully developed when they leave the bone marrow. They have to travel to the thymus. So then the secondary lymphoid tissue, as I mentioned, is where the immune responses really happen. And those are specialized structures that are associated with the tissues. And they have cool acronyms like SALT. So this is just skin-associated lymphoid tissue. So essentially, there's basically diffuse lymphocytes in the dermis and epithelium. There are skin-specific dendritic cell types that live in different layers of the skin. And basically, each specific tissue has its own associated lymphoid tissue. So SALT falls within the whole category. So SALT is one set of secondary lymphoid tissue, MALT is another. So those are associated lymphoid tissue. That's bronchial, gastrointestinal, nasal, and urogenital tract. So each epithelial surface that's exposed to the environment has its own associated secondary lymphoid tissue. So this is a cross-section of lung. These thin spaces are alveoli where gas exchange takes place. And this is a bronchus, or a main air, where air comes in to be distributed to the alveoli. And you can see there's a highly organized or somewhat organized structure of lymphocytes clustered around it. So these are located in places where the antigen or pathogen is most likely to be encountered, like junctions. Okay. So what's in that MALT? There are mature lymphocytes, so lymphocytes that were educated in the primary lymphoid tissue and recirculate. There are gamma delta T cells, which we'll talk about later, which are cells of the adaptive immune system that behave like cells of the innate immune system. There are dendritic cells and macrophages, so these are the antigen-presenting cells. So these are constantly surveying the tissue and recirculating to the lymphoid tissue to present antigen to the adaptive immune system. There are natural killer cells and NKT cells. So the organization depends on the tissue. In the skin, they're discrete cells. They're not really specific structures like the MALT. The MALT has loosely associated tissues like I just showed you. And then there are some specific tissues that have a very specialized structure like the tonsil, Peyer's patches, and the appendix. So this shows how MALT is organized in the Peyer's patches of the intestine. And you can see there are very specialized villi, essentially, that are tasked with sampling the antigens that are within the lumen of the gut. And so there's a traffic of leukocytes from this epithelial layer and associated antigens into regional lymph nodes, and that's where all those antigen presentation happens. And if there's an antigen to respond to, there's a germinal center. We'll talk about that in another class. Okay, so the secondary lymphoid tissues we've talked about so far are specific to surfaces that are exposed to the environment. There's a systemic system of lymph circulation that connects the rest of the lymph nodes in your body. So this is a separate set of capillaries that drains extracellular fluid. So in general, you understand that the circulatory system leaves the left side of your heart, goes into the arteries, passes out into capillaries in some tissue, and then passes back into your venous system, back to the right heart. During that process, some fluid is lost, and that comprises lymph. And that lymph drains into a separate capillary system of the lymphatic system. And so there are capillaries that drain that extracellular fluid, and with it, pathogens and antigens are picked up. And so that basically flows, doesn't have a pump like the heart or the cardiovascular system, but basically the constant production of extracellular fluid creates a positive pressure. And so there's unidirectional lymph flow with valves, because a lot of it's against gravity. And it all condenses into two main vessels, the right lymphatic duct and thoracic duct, that empties back into the venous circulation. So this diagram basically shows an example of the main lymphoid drainage systems for the lower limbs, which basically condense into a set of lymph nodes in the abdomen, and the upper limbs, which condense into a set of lymph nodes in your armpits and your neck. So when you have a systemic infection, or even a regional infection, you know you can get swelling of lymph nodes in your groin, in your armpit, in your neck, depending on where your infection is. And that's because there are adaptive immune responses going on in those lymph nodes through which the lymph is draining. So it's not used very much anymore, but there are actually medical procedures called lymphangiograms where those capillaries can actually be diagrammed. So the process is to inject blue dye in between your toes. That drains into the lymphoid system, and then using that dye to find a lymph vessel in the foot, they then infuse a radioopaque dye so they can take CT or X-rays of the lymph node. So you can see this is a pelvis. This is someone's pelvis with their safety pin holding up their Johnny, and these are the lymph nodes in the pelvis draining up into the abdomen. And this is a picture of someone's lower leg undergoing a lymphangiogram. You can see the radioopaque contrast passing up through the leg. Okay. So you're producing all this fluid into the tissue, and that all drains into a regional lymph node. So there are two ways for those antigens to get to a lymph node. Either there's an antigen presenting cell out there in the tissue that grabs onto something and takes it to a lymph node, or the antigen or pathogen filters into the lymph and is filtered into the lymph node. So there are antigen presenting cells always in the lymph node picking up, sampling the lymph. And so this is where the lymphocytes encounter antigen during an immune response, whether it was brought there by an antigen presenting cell or filtered there by the lymph. They're organized, the lymph nodes are in chains along those lymph vessels, and the multiple vessels basically enter a central cortex, and the lymph passes out from the cortex into the medulla. We'll show a drawing in a second. So, and then out into a lymph duct. So basically, a whole bunch of lymph vessels come into a lymph node and are filtered past the lymphocytes and then leave in one larger vessel. So all of those small lymph capillaries are basically condensing down to one large lymph vessel. So in the cortex, there are B cells, follicular dendritic cells, which are specialized dendritic cells for the lymph node, and macrophages. And then out in around the cortex are T cells and dendritic cells. And then in the medulla, there's plasma cells. And plasma cells are the specialized B cells that produce a lot of antibodies. So when you have an immune response, the dendritic cell takes whatever the antigen is, presents it to T cells. T cells promote B cell differentiation into plasma cells, and antibodies produced. And that's just a snapshot of processes we'll go through later. So again, for example, this could be in your groin, all of these lymph vessels from your lower leg draining into the lymph node. That all that lymph goes centrally past all the lymphocytes and antigen presenting cells. If there's an immune response, then a germinal center occurs, and so this is where B cells expand and do what they do. The rest of the lymph passes out to return to the circulatory system, and there's a artery and vein to bring in lymphocytes into the lymph node. Okay, so the last important major tissue of the secondary lymph system is spleen. So similarly to lymph nodes basically filter all that lymph for antigens, the spleen filters the blood. So it's structured very similarly, except it's a filter for blood. So it's a trap for injected or overwhelming infection that reaches the blood. It's not required, except for optimal responses to some encapsulated bacteria. There are a few people that are born without a spleen. There are some of us that lose them in an accident or surgery. Probably I'm not the only one in here that doesn't have a spleen, but you can survive quite well without one. It is useful for pneumococcus and certain other bacteria, but basically, again, it's organized very similarly to a lymph node, except rather than the lymph coming in from outside, the blood's coming in through a vein and then being exposed to all the lymphocytes for filtration. I mean, through an artery, I'm sorry, before passing into a vein and back into circulation. Any questions on primary or secondary lymphoid tissue? Okay, good, we're in the home stretch. There may be some sushi left. Okay. So how do the different lymphocytes know where they need to go? So you've got T cells coming out of the bone marrow that are headed for the thymus for education. You've got B cells and T cells that have to move around the body and get in and out of lymph nodes. You've got neutrophils in circulation. They pretty much stay in circulation until they see inflammation. So there's a very complicated system of signaling that goes on for all that movement. But there are some basic components of it that we'll talk about today. So the basic thing is, if you're a white blood cell in circulation, how do you get out of circulation? So extravasation is how a white blood cell gets out of the circulation into a tissue. And it's essentially a multi-step process in which the cell has to get out of the high-speed circulation. So it has to slow down by rolling along the endothelium of the blood vessel. So essentially that results from interactions between molecules on the white blood cell surface and the endothelium. It happens best in post-capillary venules, so small blood vessels that have very low-speed blood flow. It's mediated by a low affinity of interaction of E-selectin and its ligand. And if an inflammatory signal is present, there's increased expression of integrins on the endothelial cells that help to tether the white blood cells to the endothelium. So if there's enough stimulation of the lymphocyte in that that's rolling along the endothelium, then it activates, flattens, and can leave the circulatory system. So the first part is margination, the second part is diapedesis. So the lymphocyte that gets arrested has to actually get through the endothelium and out of the blood vessel. So it sticks a pseudopod or a foot between endothelial cells uses enzymes to digest a path through the basement membrane, and then they follow soluble stimuli, chemotactic gradients through the tissue to the source of inflammation. So pathogen molecules, as we saw in that initial YouTube video of the neutrophil following a bacterium, they can follow pathogen molecules, they can follow chemokines, which are secreted by the tissue itself, and they can also follow portions of the clotting cascade. So again, like I said, if you have a wound that results in a blood clot, then some of those clotting components are released into the circulation and recruit inflammatory cells. So, I have too many word slides, so we'll have one more movie. Supposed to be sound. Okay, well, you're not getting the sound, but basically this is showing what I described. There's a locus of inflammation in the tissue that's releasing inflammatory cytokines, which causes upregulation of selectins on the endothelium, such that there's enough stimulation to cause those neutrophils or other white blood cells to stop circulating. Then the cells themselves have to activate and digest a path through the endothelium. So here's an example. This is an ear of a mouse. This is intravital microscopy showing the smear is the regular flow of blood, so most of the blood cells are cruising by. Can't even identify them individually. But these leukocytes are rolling, essentially. They're rolling along the endothelium. And then one of them up here basically stops rolling, it gets arrested and activates and then digests a path out through the endothelium into the tissue. There's another blood vessel up here. So here's some leukocytes that have already been stimulated to leave the circulation. So that, in terms of entering tissue directly, that's the process by which white blood cells leave the circulation.
How does it seal the endothelium back up?
That's a good question. It's not, I don't know the answer specifically. It's not the job of the immune system. Presumably, whatever processes the endothelium uses to maintain its integrity activate once the leukocyte passes through. Yeah. Synthesizes basement membrane and tight junctions between the endothelial cells.
I thought, like during inflammation, that the lining of the blood vessels become like more permeable,
That's
so
right.
less selective. So would it just be, it's not necessarily like breaking through the lining? It wouldn't it just be
So-
easier to diffuse?
So there's different levels of integrity to the swelling that happens during inflammation is because the endothelium becomes leaky, but those pores are basically let through soluble components and fluid. It's not big enough for cells, so it probably makes it easier for cells to get through, but they still have to basically digest a path through the basement membrane, even though, you know, when the endothelium is receiving inflammatory signals, it reduces the tight junction between the endothelial cells, so fluids and solutes can get out, but it's not big enough for the cells to get out without undergoing that specific process of adhering diapedesis. Okay. So what we just looked at was basically an innate immune response or neutrophils and monocytes leaving the circulation to the site of inflammation. T and B cells are unique in that they constantly recirculate. They don't only leave the circulation for inflammation, but they circulate through all the lymphoid tissues. So you can think about the fact that we have a fixed set of lymphocyte specificity. So how is the lymphocyte going to know whether the molecule that it reacts to is in your foot or in your head? And the way that that is solved is basically by constant recirculation of lymphocytes through the lymphoid tissues. So to increase the chance that a lymphocyte and its specific antigen interact, the lymphocytes are constantly moving through the lymphoid tissue. And it's regulated by those very specific postcapillary venules called high endothelial venules. And they have a constitutive expression of lymphocyte-specific adhesion molecules. So that's a place where just lymphocytes are recruited out of the circulation to enter the lymphoid circulation. And so they're circulating through the lymphoid system and through the lymph nodes essentially constantly. So if there's no antigen, no inflammatory signals in that lymph node, they just continue down to the lymph duct and go back into circulation. If the antigen's present, then they are retained and they undergo an immune response.
Since the circulation, as you said, is by like positive pressure, it does not need any energy. It's like, is that does it need any energy?
Well, I mean, there's no, every specific movement requires energy. So it requires a functioning circulatory system so that there's a continuous extravasation of fluid. There are valves to make sure that that moves in a particular direction. And similar to the venous system, when you flex muscles, especially in your lower limbs, those vessels are compressed. So because of the valves, the fluid can't flow backwards, it can only flow forward. So there's not an active pump running the lymphatic system, but it has the same sort of passive pumps that the venous system has. Good question. Okay, so there's a whole bunch of very specific interactions that tells lymphocytes where to recirculate. So not only do they recirculate specifically in the lymphoid tissues, but they're actually tissue-specific addressins that target some lymphocytes to skin versus gut versus different places. Again, not important to memorize, but just the two important concepts are lymphocytes are constantly recirculating, but there's also a specificity for which tissues they go to, and there's also a specificity on the recirculatory patterns of memory cells, so cells that have already undergone an immune response versus naive cells. So when lymphocytes undergo an immune response, if their first immune response is in the gut, the memory cells from that immune reaction are programmed to be more likely to recirculate to the gut, because the likelihood is that the next exposure to that same antigen will be in the gut. And that's incompletely understood. Okay. So, last 10 minutes. Okay, so we've talked about cell surface molecules that the lymphocytes express at the endothelium expresses to help control recirculation. There's also longer distance communication, not just cell-cell interaction, but the communication of soluble molecules, so less than a whole cell, that diffuses to affect another lymphocyte. So these include cytokines and chemokines, PAMPs, so those are the pattern recognition molecules that bind to PRR, so those could be bacterial products. Immunoglobulins are antibodies, and adhesion molecules. So these are all the receptors on these are the receptors on the plasma membrane that can sense all those different soluble factors. So there are multimeric formations that increase the regulation. In other words, it's not a situation of one molecule and one receptor, but a complex system of combined receptors to maintain specificity. And usually there's a cytoplasmic or extracellular portion that initiates an intracellular signaling cascade. So cytokines are particularly important for that communication. They are short and long distance, resulting in short and long distance communication between leukocytes. They're small proteins that are secreted primarily by leukocytes, so a few are made by tissue cells, but mostly they're made by leukocytes. And they are important for regulating the innate and adaptive immune responses. In other words, are you making an antiviral response or an allergic response or an antiparasitic response? Some cytokines are anti-inflammatory or counter-regulatory, so they're not all just pro-inflammatory. So you'll see in your book there are, I think the cytokines are numbered up into the 50s now, so we're only going to learn the function of a few of them specifically throughout the course, but this slide just exemplifies the fact that there's not just a one-to-one relationship. There's not one cytokine that does one thing, but it's the concentration and timing of combinations of cytokines that result in immune responses. So as we've talked about, naive T cells come out of the bone marrow, recirculate to the thymus to be educated. So then they are a naive T helper cell waiting to undergo an immune response. If they see their antigen and perform an immune response, the cytokine context in which they are determines what kind of T cell they differentiate into. So there's at least the 3 types described here, Th1, Th2, and Th17. There's many more. And what you can see is that there are 3 cytokines that lead a naive T cell to be a Th1. There's more that lead to Th2. There's a complex order that come to Th17. Don't need to memorize any of these things, but just keep in mind that it's an incredibly regulated system that controls not only whether an immune response happens or not, but what sort of immune response happens. And that's partially regulated by cytokines. So here's a system where there are many cytokines that allow for combination. There are other cytokines, typically more to do with the innate immune response, where one cytokine can have many effects. So there are a couple of key molecules that are central to the innate inflammatory response, like TNF. So the prime molecule in inflammation that causes that upregulation of adhesion molecules in the endothelium is TNF. So you can see where one molecule, TNF, causes adhesion to the endothelium. It activates leukocytes, so it helps them get out of circulation and causes them to produce cytokines. It has effect on structural cells as well that undergo inflammation. And then it also has systemic effects, so it mediates a lot of the feelings you have systemically, physiologically, when you have an infection. So here's an example of one cytokine that can have many effects. So if you want to intervene medically, therapeutically, you could theoretically have a much more specific effect by targeting one of these cytokines. You could have a much broader effect by targeting something like TNF. And so targeting TNF is actually a therapeutic approach that has been used very successfully for arthritis and other inflammatory diseases. So TNF, as described in the previous slide, has many effects, mostly produced by macrophages in inflamed tissue, either infected or inflamed, activates endothelial cells, activates leukocytes, leads to production of destructive enzymes. So has a very important role in the knee, where basically an inflammatory autoimmune response leads to destruction of cartilage. You get bone-on-bone interaction and loss of function in rheumatoid arthritis. So there are now at least 4 approved drugs for targeting TNF. These are all large molecules. One of them is a soluble version of the TNF receptor itself. Some are antibodies against TNF, and they're all very effective in treating rheumatoid arthritis. What I've shown down here is that in studies of basically 5,000 patients, it's been clearly proven that taking anti-TNF increases your chance of a serious infection by about 2-fold. So not surprisingly, the immune system didn't evolve TNF to destroy cartilage, it evolved TNF to fight infection. So in deciding whether you're going to take an injection of anti-TNF to help with your arthritis, the benefit has to be weighed against increasing your chance of a serious infection by 2-fold. Now it's important, while it sounds scary that you increase the chance of infection by 2-fold, you have to realize that there are basically, out of 3,500 patients that took the drug, 126 serious infections, whereas in 1,500 patients that took placebo, there were 26 serious infections. So even though the odds were increased by 2-fold, it was a relatively low number of events. So for those of the 3,500 patients that didn't get infections, they received the benefit. So it just highlights the fact that there's a risk benefit to any medical intervention, and in most cases, intervening in the inflammatory process will increase the chance of infection. And this just highlights there are many other mechanisms that are in clinical development for treating arthritis, which include antibodies against IL-6, antibodies against T-cell receptors, as well as oral drugs to inhibit signaling molecules. And this one, tazocitinib, was approved last year. It's the first new oral drug for rheumatoid arthritis in about 20 years. That's it for today. Happy to stay as long as anybody wants. Or if you want to get sushi, communicate with me by email. I won't be on campus except for on Fridays, but I have Dave's email on my calendar, so it should be very easy.
Where is your office on campus?
It's here in the 313.
Which one?
313. But I won't be there unless you make an appointment.
Okay, but would you be there like at 11 or?
If I hear from you.
Okay, that's fine. Okay.
How are you, Cody? You used to hear I'm probably gonna take it At least you're done, and hopefully you're gonna graduate. Do you have any troubles getting nice feelings? I like just got here yesterday.
Thank you.
She got a new phone.